Hello, I'm Dana, and today I will tell you about the cohort analysis functionality within CGAP. So you can try it out by yourself by logging into our CGAP training HMS Harvard EDU. That's our training portal. You can create your username following the instructions, logging in, and navigating to the cohort browser. We have an example cohort here that includes 100 cases and 100 controls, all whole genome sequenced and randomly assigned as cases and controls and jointly called by Sentient, and then statistically analyzed by a tool named Regini, which is developed by Regeneron, and provides us with the following calculations, where you can see here all the genes and their corresponding burden, SCAT-O, or other tests, and the corresponding negative log 10 of the p-values. We can add additional genes, as you can see here, and we can also look at other masks. So we can use a mask with by filtering all the variants with CAD score lower than 15, and that's what we see here. In a selected mask, we can also look only at variants that are contributing to those p-values that are nonsense or splice sites, and we will see different variation. We can also select a specific gene list, and for instance, instead of looking at all the genes that contributed to the p-values, we can look only at 73 genes from ACMG 73. And obviously, there are significantly less genes to focus on. We can also focus on a gene name or a genomic position. But for now, let's look at all of them. And I can tell you as our secret that there are no statistically significant values here as these data suggests. But we just included as mock data those higher values for the tests in order to make it a little bit more interesting for a use case of type 2 diabetes. In our case, let's look at the first real gene, and I can now focus on gene LAMA2 by going to the Cohort Browser and looking at the gene more closely. So the Cohort Browser uses our high-glass server, and what we can see here is the different genes on the first track, the negative log 10 of the p-value of the selected test, which is the burden test in this case. We can see the selected mask, which is the CAD score mask, and again, those are changeable facets. We can look at different tests, and we can look at different variants over here. And by going back, we can just go to the gene of interest because it's quite dense to look at all the variants over here. And the variants that we see are the variants within the Fisher exact test p-value of our cases and controls. And they are colored by most severe consequence level, where high is red, yellow is moderate. We can also add the low consequence variants that include the synonymous and the splice region variants versus splice sites. And we can also add modifiers over here, and the modifiers will include intronic variants, and usually those variants are not included in our test calculation. So let's go to the gene that we mentioned before, and that will help us to focus on much smaller region. So right now we are focusing on much smaller region, and we can see here all the introns and exons of the gene, And we can also look which variants were included in the calculation of the burden in this gene, the burden test of this gene. And as you can see, the included variants are highlighted. So if you notice, whenever I hover over this p-value, I can see that many of these variants are actually not included in the calculation. And the reason for that is that we are including only variants with a CAD score, which is higher than 15. And most of the modifiers and low consequence impact level have quite low CAD score, so they were not included. And if we exclude from our visualization the low and modifier consequence levels, and we click here, we can see that not all the moderate variants were included. And when we go, for instance, to this variant, we can click and we can see that there are variant details in terms of the position, the occurrences with cases, controls, and NOMAD version 2 and 3. We can also look at the variant association test statistics, and we can look at several annotations from the predictors, including GERP, SIFT, Polyfen, Splice AI. And for the COD score, it's 13, and therefore this variant was not included despite being a missense variant with a moderate consequence impact. So I can close that. And now just want to say that when we hover, we not only see the selected variants on the lower track, but we also see some summary statistics for this specific gene. When we click, we can see more expanded view of these statistics. So we can see the different masks and their corresponding p-values in those different tests that were included in the Regini calculation. We can also look at the variants that are included in each mask. And when we have access to the sample level information, we can see which samples are actually having these variants and have been included here. In this case, since it's training data that's available for everyone, the sample level information will not be available, and therefore, also when we go to the variant, we will see that sample level information is not available, and therefore we cannot go back to the sample and check all the information that we would like about the sample. However, we can look at the information about the gene, and by looking at this gene and transitioning to the Annotation Browser, we can look at the different annotations for this gene from our annotation database. And specifically, we are looking here at 3 genes, and we can look at all of the transcripts that are included. We can zoom in a little bit, and by zooming in, focusing more on the different annotations that we can see here. So beyond the transcript track, we have the ClinVar variants track, and we can see here all the pathogenic, likely pathogenic, uncertain significance, likely benign, and benign submissions to ClinVar. And we can zoom in a little bit more to learn more about that and focus on a specific exon, for example. So here we can go to information that would provide us with information about a specific exon. And here, after zooming in enough, you can actually look at every amino acid, corresponding variants in ClinVar, and the actual conservation status among those orthologs at the nucleotide level. And we can see, for instance, that there are variants that are quite conserved, and there are variants that are less conserved, like this one. And we can also see the track for Nomad that shows us the allele frequencies of those variants within Nomad. So can definitely get a lot of useful information from this annotation browser, and then come back to the cohort browser and focus on additional genes of interest, additional gene lists if we are interested, as well as selected control groups that could be either the verified control, which I'm reminding you were randomly assigned in our training portal, or you can control looking at the Nomad version 2 data or the Nomad version 3 data. You can play with your minimal CAD score if you would like. You can look only at variant with CAD score that are higher than 20, and you see that many of the variants are now disappearing from the visualization. And you can keep visualize and explore your data as much as you would like using our training portal. And we would highly appreciate your feedback or your questions by your submission using this functionality over here on the top right. I hope you will enjoy playing with the data, and please let us know if you have any questions. Thank you.